And now we have the final speaker of the meeting. I can feel the excitement in the room as we think about it being Friday afternoon. But we're fortunate to have Arturo Casadevall here. He's Bloomberg Distinguished Professor of Molecular Microbiology and Immunology and Infectious Diseases at the Johns Hopkins Bloomberg School of Public Health and Johns Hopkins School of Medicine, and the Alfred and Jill Summer Professor and Chair of the W. Harry Feinstein Department of Molecular Microbiology. Yes, there are a lot of words, and I was trying to read them all. And he's an internationally recognized expert in infectious disease research with a focus on fungal and bacterial pathogenesis and basic immunology of antibody structure function. And he's here to tell us about the virulence of pathogenic microbes. So thank you.
Thank you very much for inviting me to this wonderful meeting. Thank you very much for staying for the last lecture. I have never been an anchor speaker before, but there are some advantages with that you get to listen to the whole meeting and try to think of some ways in which you could connect with some of the very diverse talks that we have heard. So I thought that one of the things that comes up is a Joint Genome Institute question in the issue of virulence, so that you can potentially apply this to any of the other big phenotypes. How come we go from genetic knowledge to virulence prediction? And in particular, imagine you knew everything from a microbe and a host. How much can you really predict? And I think that's a very deep and fundamental question that I will try to address. But I also want to address some other existential questions that are fun, and this is why I went into science. Why are we here? And I don't mean to say because we're in a meeting. Why are we so hot? And I mean temperature, not other ways. Why do we eat so much? Many of you had 3 meals today. I'm going to have 3 more meals before you go to sleep. And why are mammals the dominant large animals? And I'm not going to stress you out. I'm going to give you the answer, and I promise you, I'll come back at the last slide, and this will all be put together. The single word answer for all these very deep questions is fungi. I promise you this will make sense. So I'd also like, when you talk about any area, you should look at the etymology of words. They can teach you a lot. I'm talking about virulence, so the etymology is from the Old Latin. And it enters Old English around the 15th century, and it was beginning to be used already in reference to wounds full of poison. By the 19th century, it became associated with the germ theory disease. So what is the germ theory disease? The germ theory disease replaced the miasmic theory of disease. Prior to the late 19th century, diseases were thought to be caused by foul airs, foul influences. Think about the word malaria, bad airs. And there's a representation of cholera spreading through the air. And the big proponent was Girolamo Fracastoro, who was famous enough to be painted by Titian. And we haven't gotten rid of miasmas. Whenever there is a natural disaster, you still hear about bodies in the street. They need to be buried because they spread disease. I'm sorry, they may be unsightly, they may be malodorous, but they're not something that spreads disease. So the germ theory disease was happening in about 30 years. These are momentous years, and was associated with these individuals. And basically, at the end of the day, there is nothing more than the association between a microbe and disease. And this was tremendous at the time because it provided a lot of the mechanistic, causal types of looking at disease that have made medicine a very successful field. I want to point out to you something that is not well known. One of the giants of this time, Virchow, did not accept the germ theory disease. This is the individual, to put his contribution is, he associated organ failure with disease. It was another one of those great landmarks in the history of medicine. But he believed that germs were found in pus because they rather go to the pus. They prefer to be in pus. He would not give up the idea that infectious disease was caused by poverty because it conflicted with his social views. Why am I telling you this? Because we live at a time of a lot of polarization. And people often get totally worked up when other people don't agree with them. And I think it's important to just be gentle on people and realize that if this set of giants couldn't convince one another, it's all right, things will work out, keep going. So the germ theory of disease, in fact, changes a lot. What it changes primarily is mortality. There on the left, you have all-cause mortality, and look how it drops dramatically, except for that sharp increase in 1918, which is the flu epidemic. And so to put this in perspective, had the germ theory of disease not happened, a third of you would not be here. Simply, you would have not made it out of childhood. All families lost children. The social structure was tremendously different. Epidemics came, they killed people with no warning, and will go forward. And today, we live in a world where that doesn't happen as often, in particular because of this enormous intellectual advancement. And you can see on the left, on the right, maybe your left, is the human population takes off dramatically. But by the late 20th century, there is a crisis. And the crisis goes as follows. In the germ theory of disease, there were microbes were classified as being pathogenic or non-pathogenic. By the 1950s, things were happening. Organisms that were not pathogenic were beginning to be pathogenic. And what happens is that medicine is advancing. It's generating chemotherapy, steroids, antibiotics to change the microbiome. And you're beginning to see organisms that were previously not pathogenic now causing disease. By the 1970s, people try to save this idea that it is the microbe that is causing the damage by creating the concept of opportunism, which tries to explain how known pathogens cause disease. And by the 1990s, known pathogens have become pathogens. What do I mean? I mean that if you go into a hospital today, here in San Francisco, and you go to the clinical microbiology lab and you look at what they are recovering from individuals, that microbes are very different than what would have been causing disease in the year 1900, for example. And microcentric theories cannot cope with these changes. Now, and consequently, we have a messy lexicon. So Candida albicans, everyone in this room has Candida albicans, is a commensal in most of us, but it can be a primary pathogen in women who get vaginitis or an opportunistic pathogen in AIDS. Whenever you see a messy lexicon that tells you that there are conceptual problems in a field, this is basically different names for a microbe. The same thing you can say for pneumococcus aspergillus. So in the late 90s, I was an assistant professor at Albert Einstein College of Medicine, and I was assigned to teach a course in microbial pathogenesis. And I recruited my good friend, Lysiane Porowski, and we put together a course. And I always say to people, if you have an opportunity to teach, always say yes. Because when you teach, even though it's a lot of time, and even though it takes you away from your research, and it's got all those issues, it's a time in which you process information very differently. When to teach it, you have to do it. And I will tell you that by 1999, our class notes had new thought in it. And when the question was how to get it out. And so one of the problems today is that if you have new ideas, new thought, it's very hard to get it published. Because where do you put it? So we began to hide it in mini reviews. So you write a mini review, you convince them to do that, and then you put your new ideas in there. This is a problem with current day biology. Current day biology, and in fact, has become untethered to theory. And in fact, many people basically do work in the absence of theory. And this is something that hopefully will fix itself. But we have, and this is an example, we were able to redefine the basic concepts of virulence and pathogenicity. And this is the main contribution. The main contribution is that all these interactions, and I'm talking about animals here, I realize that this may or may not apply to the plant interactions, is the damage. What we really care about is host damage. And the damage occurs as a parabolic function of the host response. People get into trouble when they mount too weak an immune response or too strong immune response. And the big intellectual contribution is that all these states, commensalism, colonization, latency, things like carriage, all these things are in fact continuous. They're not different. And if you drag down below the x-axis, then you're into the realm of symbiosis, which will be negative damage. And therefore, this has some profound implications if you begin to think this way. One of them is it shifts the debate from pathogen to non-pathogen to the outcome of the interaction. In our mind, there are no pathogens. There are only microbes and hosts. You put them together and you get an outcome. That outcome could be symbiosis. That outcome could be disease. Now, to call the microbe a pathogen or a symbiont, you're giving it a trait that is not its own, because no microbe can be defined as such in the absence of a host. Virulence is only one outcome of the interaction, and these outcomes are continuous and different only in the amount of damage to the host. This is a very, very different view than people, for example, study one of these, to try to place the microbes in this categories. And it is flexible. In 2015, we were able to add the microbiome to this. And again, we're still writing it in many reviews, because thought is still very, very difficult to get published. And I often wondered, you know, if Darwin had written The Origin of the Species today, where would he have published it? The guy had no mechanism, right? So where would he have published? Well, it turns out that he published it in a book, but this just gives you an example of the problem that we're facing. In my mind, thought is just as important as data and experimental data, and the two, we need to find a way to balance things more. That also raised the question, well, what is a host? It turns out that hosts are other living creatures. We don't call a rock a host. Why don't we call a rock a host? The rock has a microbiome. Well, the reason is that a rock doesn't respond. The plant, the worm, the human out there all respond and creates a new novel state, which is the state of commensalism, symbiosis, disease, etc. So a few years ago, they were asked to write this view in which arguing that the term pathogen should be ditched, because it's in fact, it's a term that gives a microbe something that is not its own, and that it may be affecting how you think experimentally, and it may be leading people to the wrong types of thinking. Imagine, for example, if the words, thinking without words, it's very hard. You need to have words to think. And if the words are not quote unquote right, your final output in thought may not be correct. So one of the things I decided to do was, could a mathematical formalism be developed that would account for both the host and the microbe, that also illustrated the continuity of interactions? And began playing with this and came up with something called the pathogenic potential. The pathogenic potential is a very simple formula. It looks at the fraction symptomatic divided by the inoculum, multiplied by 10 to the m. M is mortality, and that 10 enters only because mortality is a lot more dramatic than diseases that don't have mortality. If there is no mortality, the m becomes 0 and that reduces to 1. But here is what's important. I went through the literature and dug up all those animal pathogenic organisms. And then I calculated the pathogenic potential for mice. And what you can see is that it's largely continuous. There is no place in which you could put a boundary and say, these are pathogen and non-pathogens. If you increase the inoculum, you can always take down the host. If you make the host immunosuppressed, or if you change it, you can always change the virulence of the organism. So this is now keeping the microbe constant and changing the mouse strain. And what you can see is the pathogenic potential can change by as much as 100-fold. I'm glad to see that people are beginning to use it. Instead of using the term virulence, the term virulence is in fact a very problematic term, because it is always relative. We always measure it relative to something else. The mutant was less virulent. This strain is hypervirulent. It has, whereas the pathogenic potential really allows you to do this with very few numbers that you can gather in a typical experiment. So now I'm going to focus to fungi, because they teach us new lessons on the problem of virulence. So the fungal kingdom, I mean, there were some great fungal talks here, and I compliment the speakers, as will not come to surprise to them, it's enormous. It includes the major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Fungi are currently devastating entire ecosystems. The white nose syndrome, the frogs are in trouble, salamanders in Europe, the snakes in North America. But none of you worry about fungal diseases. The majority of humans worry about malaria, you take prophylaxis. You worry about Zika virus. You worry about tuberculosis. You may worry about, something like athlete's foot or maybe a superficial fungal infection. But most humans don't worry about dying from fungal diseases. That, to me, is fascinating because if you were trees, you'd be really afraid of the fungi. If you're insects, you're really afraid of the fungi. So what makes you so special? Well, in fact, there are very few fungal species that are pathogenic for humans. And the ones on the left tend to cause disease primarily when there is a problem in immunity. The ones on the right, we tend to get infected, tend to be asymptomatic unless one is immunosuppressed, or only one gets a massive inoculum, for example, goes into a cave and gets exposed to a lot of histoplasmosis. But fungi are devastating entire ecosystems. I mentioned this before. You can now see it associated with pictures. And again, I pose to you the question, what makes mammals so special? So John Perfect and others carried out a series of experiments 20 years ago that provides a hint. If you take a bunny and you infect it with a fungus known as Cryptococcus neoformans, which is a major pathogen of humans, I'll be saying more about it, you cannot give it a systemic infection anywhere. The bunny is 40 to 41 degrees, and the high temperature keeps the fungus, it stresses it such that the immune system rapidly eradicates it. However, you can give an infection on the skin or the testicles that are cooler, but if you give steroids that depress immunity, you end up with a dead bunny. So the emergent picture is that you basically need to have adaptive immunity plus endothermy, and you get this resistance. Frogs have great B cells and T cells, but they don't have temperature. And when a fungal infection occurs in human populations, it's often a reflection of the immune status. The AIDS epidemic was associated with a lot of fungal diseases, primarily because you lost one of your two protective pillars. You lost adaptive immunity, and temperature alone was not sufficient. The white nose syndrome in bats is an example of this phenomenon. Bats are 37 degrees in the summer, but when they winter, their temperature drops to 10 to 12 degrees. And this fungus that was introduced in the United States, probably from Europe in 2006, and has now killed 100 million bats, then they become susceptible because they lose the temperature. If you take the bats that are infected and you bring them back into the laboratory and you feed them, and their temperature goes up to 37, they're able to eradicate the fungal disease. So you need both. And again, what makes mammals so special? Well, I began working with Vince Robert in Belgium, and he works in a collection. And we asked the question, if you look at all the isolates in the collection, what is the thermal susceptibility of it? And what you can see in the top is that the majority of them do pretty well in ambient temperatures. They do pretty well up to 30. But after 30, there is a dramatic decrease in the percentage of fungal species that can replicate. And if you, between 30 and 45, for every degree that you increase the temperature, you can exclude 6% of fungal species. So Aviv Bergman, who you'll hear more about in a little while, took that data and he took the well-known cost of metabolism. This is a well-known formula in biology. And he set up both equations simultaneously, and he asked, where is the maximum? Where do you get the most protection for the amount of food or energy relative to the fungi? And the temperature that came out, I'm sorry, I didn't put it in centigrade, it's 37 degrees. So that suggests to me that this could have been coincidence. But again, it's your temperature. Your temperature appears to be very optimal for the cost of being hot and the benefit that one is getting for fungal diseases. The cost of the mammalian lifestyle is extremely expensive. That is a well-known plot in biology. It's a log-log plot, and we are at the upper end. This is what it costs to be a mammal. And mammals were a very minor group until about 65 million years ago. And then something I'll tell you about happens, and then you have the great mammalian radiation that gives you bats and gives you whales and gives you humans. And the question that I was fascinating is, how did this very energetically unfavorable lifestyle become dominant? I began to think there had to have been something that selected for this, Because in reality, well, let's go there. And I ask the question, could the explanation be in the distant past when the mammals emerge? So if you look at the bottom, land animals, the amphibians, the reptiles, the mammals, they all have deep origins they branch very, very deeply. And for most of that time, it's the age of the reptiles. And then you have the KT event and the main, which is the end of the Cretaceous. And you have the replacement of the reptilian megafauna by the mammalian megafauna. And what comes out of that is a flipping. You went from ectotherms, so individuals that don't regulate their temperature, to endotherms, which tends to be mammals and tend to be birds. And, so that leads to a question that you hear a lot about. So what killed the dinosaurs? Well, there are a lot of ideas. People used to think that it was small mammals that ate the eggs. This makes no sense at all. Neither does the next slide, but I found it on the web, and I figured I'd show it to you. It's out there. It could have been massive volcanism that opened up about 65 million years ago, what is the Deccan Trap. There is a plume that opens up every now and then, the plume that created the Hawaiian Islands. This is why the Hawaiian Islands are in a line. When it opens up, it has tremendous effects on the climate. So it turns out that this opened up about 65 million years ago, and 100,000 years later, which is nothing in geologic time, we had the asteroid that hit, that is well known to have hit the Yucatan. There are a couple of other explanations out there. I'll share one with you. This is in a very respectable journal. Somebody calculated that they could have passed enough methane to affect the climate change. I leave it up to you to think about that. There is a reference, 2012. And the next is something I found too on the web. I don't have any evidence for or against, and as a scientist, I won't comment on it, so I'll present it to you in silence. So the Cretaceous world and its end. It was a warm volcanic planet. The Atlantic was tiny. There were forests in Antarctica, and the rock hit which was about the size of Manhattan Island, hit the Yucatan. And if you go to Yucatan, to Cancun, they often invite you to go see a cenote, a place in which, they say Mayans did the sacrifices. Well, next time you go there, you should go see the cenotes, primarily because the geologic formations are found only in the Yucatan, and they are the rim of the crater on the part that is on land. The post-impact world was a pretty hellish place. You had the temperatures drop 12 degrees centigrade overnight in the planet. Photosynthesis was shut down for 6 months and as much as a decade. And you had fire, smokes, and suns obscure the sun. You could find evidence of this in the geologic record. So the reason that you had the fires is because a lot of that Earth that is kicked up into low orbit is now re-enters as flaming meteorites. And here, and the other thing that was associated with this is a major fungal, the Earth became a fungal compost. There is fossil evidence that as those forests came down, you had massive amounts of fungi. You think about it, cool, dark, decayed, dead vegetable matter. You would have had massive amounts of fungi growing, massive amounts of spores. And I began to think, maybe this is part of the connection here. Maybe it was the fungi that kept down the reptiles so that we don't have a second reptilian age. I haven't had never heard a really convincing explanation as to why we had a mammalian radiation. By the way, I drew that, and I urge you to draw your science whenever you have a chance. Doesn't take that long, and it doesn't have to be that good. But I'm really happy with my meteorite over there. You know how hard it is to get that with watercolors? Anyway, so about 15 years ago, I sat down and wrote this idea, tongue-in-cheek, and it turns out that it's out there. And the idea is that in the post-impact world, the great reptilian megafauna is probably killed by the blast. Anything at the top of the food chain would have been in deep, deep trouble. The chilling of the planet, maybe fungal diseases. Reptiles fight off fungal diseases by sunning themselves, but there was no sun in this world. So they would have been at the mercy of whatever temperatures ambient. Reptilian eggs are susceptible to fungal diseases. In fact, many fungi go right through the egg and kill the embryo inside. And there is fossil evidence that dinosaur eggs used to be susceptible to fungal diseases. So I think you can begin to put all this together with this great fungal compost. And instead of asking what killed the dinosaurs, say, what kept down the reptiles such that we did not have a second reptilian age. Reptiles need a tenth of the food you need, they reproduce a lot faster, and they should have eventually repopulated the planet. So the idea is that you went from the age of mammals to the age of reptiles as a result of a fungal filter that selected for the emergence of mammals. And this is, I gave this talk at the NIH, and somebody made this nice poster for me. And if you're interested, it's all summarized in there. I don't know if this is true. I don't know. It's, to me, I went into science because I like to think and I like to explore ideas. And to me, this is so far internally consistent. And I enjoy it, and I find a way to write it up, and I find a way to get it out there. And you can do whatever you think with it, but it is consistent with the view that the reason you don't fear fungal diseases is because as a mammal, you were here as a result of a fungal filter that long ago selected for a life form that was going nowhere because it needs too much food to get around. So what are the requirements for fungal human pathogenicity? Well, you need to be thermotolerant. If the organism can't grow at 37, it is not going to be pathogenic. And then you need to have other things to get around the immune system, things that survive to take down immunity. And one of the points that I want to make is that currently we have a lot of fungi that are probably around 35, 36 degrees that are fully loaded to cause disease in mammals. But the temperature gradient is protecting us. As the planet gets warmer, some of these fungi will adapt to higher temperatures. And I think we're going to see new fungal diseases in the late 21st century. And in fact, there is a new fungal disease that has come out of nowhere in the last 4 or 5 years, independently, in multiple places in the world, from not a monophyletic lineage as Candida auris. And the question is whether this is the first example of fungi adapting to temperature and that they will then defeat our thermal barrier. By the way, that paper on the bottom shows that you could very rapidly adapt fungi to temperature in the laboratory by training them by increasing their temperature. This company was trying to make an organism to control insects, but the insects learned to sun themselves and raise the temperature. So what they did is they got the organism to grow at 37 degrees in 6 months. So anyway, this is something that is out there if you're interested. We have written about it. We have evidence that the fungi are already adapting. This is a complex set of curves. There are Basidiomyces and Ascomyces. The only ones to focus in the Ascomyces are already temperature adapted. The Basidiomyces have much lower temperature tolerance. But if you look, these are the average temperature on the dates of deposit at a collection. And what you can see is that the average temperatures or susceptibility of these organisms in 1985 were lower than in 2015, almost as if these are already sensed the change in the world and are adapting. So I work primarily with this organism, Cryptococcus neoformans. The reason I began to work on this is because it was a major pathogen. It continues to be a major pathogen of immunosuppressed people and patients with HIV infection. We are all infected, but disease is rare, provided that you have good immunity. When immunity fails, this organism can go anywhere. It has 2 major virulence factors. It's got a polysaccharide capsule, and it makes melanin. That melanin is the same melanin that is found in your skin. So the organism poses a conundrum. It has a very sophisticated virulence strategy. That is, it goes into macrophages, and it subverts all the mechanisms of killing in ways that appear to be almost designed to do so, even though this organism lives in the soil and has no need for an animal host. And it also has many hosts. You can cause disease in plants, you can cause disease in worms, you can cause disease in bees. in insects and salamanders and you name it. So non-specificity and sophistication. How do you get that? Viruses tend to be very sophisticated, but they also have specificity. So I had two questions. How does a soil organism with no need for an animal host have such a sophisticated virulence strategy? And how do you take down all these very different type of eukaryotic hosts? So the first answer we have worked on for 20 years. I'm not going to get into it. You can just say that this probably learned everything that it needed to take down a macrophage in amoeba. Amoeba are major predators in the soil, and this organism then develops an arsenal, a survival arsenal, that when it gets into the lung and it runs into a macrophage, it thinks it is dealing with an amoeba. The second question, I'm not going to go into it today, it's complicated, but what it does is we have evidence that it knows how to hit different components of the eukaryotic physiology, for example, translation and mitochondria. And this is why it is able to cause disease in such different hosts, including, and potentially that was again learned in amoeba. We can also know that this organism is pathogenic strategies ancient. We took strains that were separate by 100 million years, put them into macrophage, and we get exactly the same outcome. So these strategies of causing disease were certainly around in the Cretaceous. The other interesting fun that we had was that now that a lot of genomes have been done and there's a lot of information on these strains, it appears that the organism emerged in Pangaea, in that area between South America and Africa, and is diffused throughout the world by a variety of mechanisms, and most recently by the fact that humans move pigeons around and the pigeons are infected and they carry it with them. So we published this, and somebody who was a lot more smarter and sophisticated than we were took the same map and was able to explain some of the major conundrums in phylogeny. Why are the isolates from Spain similar to the isolates from Asia, for example? And you can see that the VG1 type is in the original, when you superimpose in original Pangaea, was kind of connected. And this kind of thinking, that is, try to think in the temporal dimension, could give you new hints of where to go. And in fact, you can then generate a lot of hypotheses. You can go to the most ancient place, and you could look now for hybrids in that area that would be proposed to have happened, whereas those away from the center are well expected to be different. So I want to get to the question is prediction possible? And I'm going to give you three independent vignettes. One is new unexpected roles for so-called virulence factors. I'm going to show you that things that appear to be well-designed for causing virulence or for something else can work in ways that are unpredictable. The second one is going to talk about the dynamics of the macrophage cryptococcus interactions and the dynamics of host-microbe interactions. So new roles for all virulence factors. We're going to focus only on urease and melanin. So urease is a simple enzyme. It breaks down urea. And if you put it in urea agar, you get this beautiful pink because it becomes basic with an indicator. This is being studied by many labs, and it's been associated with brain invasion. So the question was, is that all it did? Well, it turns out that urease is nothing more than a nutritional enzyme. It is used by the organism to get a nitrogen from urea. It cannot use it as a carbon. And then when we analyzed it, we found that the urease in a macrophage hydrolyzed urea to ammonia. It changed the pH in the phagosome, and then it had all these downstream effects that were quite unpredictable. It changed the phagosomal pH. It correlated with phagosomal leakage. It affected the leaky material, then triggered cell death in the host cell, and is an example of how nutritional enzyme put in the right place where there is urea gives you a set of emergent properties that could not have been anticipated even if you knew. The second one is a new role for melanin. So melanin is found in many pathogenic fungi, many pathogenic organisms. It serves to protect the microbe against a variety of insults, including, for example, of oxidative fluxes. It gives fungi rigidity in the cell walls, etc. So we graph the color of organisms in a collection. This is the same collection. We're still working with the same individuals. And originally I thought that, you're more likely to have pigment if you're near the equator. That is, that melanin may be protecting against sunlight. And it turns out that it was the complete opposite. The pigmented organisms were past the tropics, primarily in Antarctica and in the Arctic. So Cryptococcus has a Laccase that allows you, depending on the substrate you give, to generate colors. You can generate yellow cultures, green cultures, etc. So we began to see the effect on color on the absorption of sunlight. And the melanotic organisms heat up very rapidly when exposed in sunlight. So this was fun experiments. People ran to the top of the building, put the Petri dish out there, and with a thermal camera, you could rapidly see within minutes that these things will go up many degrees. Here is a more interesting experiment. The Cryptococcus was using, in the cold room, the melanized organisms were capturing the light, raising their temperature a few degrees, and surviving in ways in which the non-melanized weren't able to. These are the black mountains of Antarctica, which is the black is supposed to be melanin from a fungus, and it's a lichen, and an alga association. And I will bet you, even though we have tried to find the data, that these organisms are capturing heat through the melanin in that situation. More problematic is the fact that when glaciers begin to melt in the sinkholes, they often have black organisms that grow into them, and they set up a positive a cycle by which those holes get hotter and hotter and lead to the melting of the glacier. This is lichens and mosses above the tree line in Mount Washington, and you can see chlorophylls and you can see melanins. These are pigments that function in virulence and also in the environment are functioning for light capture. I'm going to talk to you about chance in the phagolysosome. And yes, I drew that. I had a lot of fun drawing it. You see macrophages, and you see that they're playing with cards, and the cards are different microbes. So before I do this, I want to tell you a little bit about my own issues, okay? So if I lived in the ancient world, I would have probably be part of the cult of Pythagoras. The cult of Pythagoras was a mathematical cult that believed that everything in the world is mathematical. And they were remarkable in antiquity because they included women. But they saw the world as if you understood the math, and obviously there was like Pythagoras, which gave us the Pythagorean theorem. Pythagoras, this is a painting in the school of Athens, is notorious because you see the tetractys, which is the same as Pascal's triangle, which is their major symbol, but you see that there are women near him. And again, it was a very different type of sect in a world that was very misogynist. But the view is, people talk about mechanism. There is a deeper level, and that deeper level is the mathematics of the system. And the mathematics of the system will tell you the degree on which anything is predictable. So I have to give you Dynamical Systems 101. This is an incredibly complicated topic, but let's just say that I got it to one slide. And a dynamical system is a system in which a function describes a time dependence of a point in geometrical space. There are two major types. Deterministic. That's a system in which if you know the conditions today and you let it go, you can know the conditions into the future. You're able to predict where things are at. And there are systems that are stochastic, in which is randomness come in. For example, the best will be Brownian motion, in which the different fluctuations of atoms gives you movement in very different areas. I want to point out to you that chaotic systems are deterministic. The main problem in chaotic systems is that they are very sensitive to the initial conditions. And because of that, they are essentially not reproducible, because you can often get back to the original conditions. Chaos theory is the example of the butterfly beating its wings in one country and setting up a tornado in another. The idea is that small changes can propagate tiny perturbations in initial conditions, resulting in wildly different outcomes. They've been discovered multiple times, And it's been only recently, I would say in the last half century, that many mathematicians have begun to work on this. So detecting chaos. Actually, this is a work in progress. And what we needed to do is work with Aviv Bergman, and I'll tell you a little bit more about him, to analyze the mathematical data that we're generating and ask the question is, what are the dynamics of the system? Are we dealing with stochastic systems? Are we dealing with chaotic systems? And he is a mathematician at Albert Einstein, so we generate data very similar to the data many of you are generating, mathematical, and when you analyze it, and you can analyze it by two major ways. One of them is an ordinal pattern analysis, in which you look for all the combinations, and you ask the question, is one not found? That will be a forbidden, you're looking for a forbidden event. The other one is you do a power spectrum analysis, and you see how the decreasing exponential, the shape of it, that will give you insight into whether it's a chaotic or a periodic system. One of them looks like a staircase. The other one looks like a continuous function. So this is just what I just said. When you look at the butterfly effects, you're looking for all the possibilities in that mathematical data set. And you're asking the question, are there any missing patterns? And what you see is that whenever the curve drops into the bottom, that will be an example of a forbidden pattern. When you find that, you're dealing with a deterministic system. And then you use the other analysis to know whether you're dealing with something that has chaotic signatures. So measuring the pH of phagolysosomes, straightforward in the cell biology world. You take macrophage organism, in this case Cryptococcus, you put a fluorescence label on it. This basically fluorescence is different with pH. You draw a curve, and you begin to measuring thousands of these events. And you ask the question, what does the data look like? Well, this is an example of looking at 4 hours of thousands of these events. Every phagosome is different. Every phagosome is a struggle between the microbe and the host. And you can see is none of those curves go to the bottom. So we could find no evidence that this was a deterministic or chaotic type of system. Instead, if you look at beads can't do anything to the phagosome. They're inert. You get a beautiful type of distribution, which is a normal distribution. It's telling you the system is stochastic. So the macrophages are acidifying macrophage containing identical particles to a final pH that differs on stochasticity, probably the number of pumps and things that happen in the formation of the phagosome. So then we looked at Cryptococcus, and when we put Cryptococcus in, the pH distributions approximate normality, but if you look at the top are the beads, and those funny signs, size means that they're normally distributed. The Cryptococcus are not. The Cryptococcus are fighting and are changing the pH, and they're changing it in a way that is away from normality. And they do so by the urease I told you about. The capsule enlargement creates a big space. Things get diluted. The phospholipase damages the membrane, so you begin to get stuff from the cytoplasm coming in. And you could have glucuronic groups in the capsule that buffer the pH. So what you see is that the macrophages are generating a normal distribution because they don't know what they eat. So they're hedging their bets to try to control the organism. Batch hedging is very common in biology. It's taking an insurance into the future. This is from Wikipedia. You can Google it. And for example, so many birds lay different type of eggs. And the little egg may not be very suitable for most years, but in a year in which there's a drought, that little egg may give a greater survival advantage. So what you see is bed hedging in this case at the cellular level in a macrophage. So these are complex curves, and what it's telling you is that when you analyze this distribution of the macrophages, you see that in fact the bed hedging that they do is very similar to some of the microbes. Oops, that slide is not there, but I got rid of it. But here what you see is the closer you are to the bottom, the better it is for the macrophage. And then what you can see is that the three in the bottom can modify their pH. the ones and the other ones do, and they move the bedding away from what is best for the macrophage in order to succeed. So at the heart of a host-microbial interaction, host-microbe interaction in the animal is the phagocytic cell. And at the heart of the interaction is going to be what's happening in that phagosome. And it is stochastic. So it means that there is a random element entering in which the macrophages are bedging their heads by diversifying how they acidify their vacuoles in order to inhibit the majority of the types that they do so. But they often don't get it right. And in those types, they may lose the battle with that organism. So we wanted to look into chaos. And the first early peek into chaos was about 10 years ago. We published a paper in 2014 in which Garcia Solace began to infect Galeria. Galeria are the wax moth. And if we were looking in a not very sophisticated way, had this been chaotic, oops, I gotta go back. Can I go? Yeah, I can go back. That curve would have been away from the line, and it isn't. In fact, we could find no signature of chaos. Now remember, just because you can't find something, it doesn't mean it's not there. So we were convinced that our gut feeling was that there had to be chaotic signatures in this type of interactions. And Aviv Bergman set up the new search for chaos, and this is doing the experiments very differently and analyzing it. Now you see that some of them reach the bottom. They become forbidden ordinals. This is Aviv's work. I credit him with this because he had to figure out how to take survival data to analyze it in a way to find the chaotic signatures. Nicole Broderick has done it with Drosophila, and we have gotten C. elegans data. So in C. elegans, in Galeria, and in Drosophila, we have preliminary evidence. By the way, this is all unpublished. I figure you should hear something that hasn't been unpublished. We're writing it up now that this is chaotic. This is at the deepest, most fundamental aspect of the interaction. It has tremendous consequences on how you think about a system. What is the reproducibility in a system? Where can you push it? And how can you perhaps mine some of this? So I'm going to end by some thoughts. Virulence is an emergent property. You take two entities, you put them together, and you get new states. So emergence is a fancy way of saying something that Aristotle said on the 4th century before the Common Era. The sum is greater, the sum is greater, the whole is greater than the sum of the parts. Here are emergent patterns. Sand formations refer to emergence. If you know all about the physics of grains of sand and Bernoulli's equations of flow, of wind, you cannot predict those patterns. You may be able to explain them afterwards, but you cannot predict them. You certainly cannot predict that funny pattern that happened in that other sand dune. So if you have an emergent system, two things flow. It is not reducible, and it is not predictable. So in my area, infectious disease, microbiologists tend to torture the microbe and hold the host constant. Immunologists torture the host and hold the microbe constant. This is the only way to get really clean results so they can get them published in places that are desirable. But I would argue that both are basically forcing experiments that don't reflect the reality. Even though they can be very informative in some ways, there are problems, fundamental thought problems, on how we're approaching some of these very complex systems. So on prediction, it's going to be very difficult because virulence factors manifest new emergent properties. So if you know the sequences and you know all the organisms and you know the genes, some of those genes, when put into a new environment that is a host, may function in ways that were unpredictable based on what was the previous known roles. At the cellular level, you see stochastic interactions in the phagosome. That means an element of randomness, that the outcome is going to be determined by factors that are random, that are not going to be able to be predicted. And host-microbe interactions show chaotic signatures. Chaos is deterministic, but you cannot go back and set the conditions identically. And this may be why no two host-microbe experiments are ever identically reproducible. because it is, you can't even, if you could do so, you could reproduce it, but the fact is the variability is beyond what can do. But I would say do not despair. These are the prediction of weather forecasts. And weather has all those traits. You can find weather is an inherently chaotic system. And what you can see is that with experience and computational power, things get better. So I do believe that as the century progresses and the kind of work you're doing and you're generating some of the data that will then be synthesized in the realm of experience, the realm of probability, statistics and all that, that it may be possible someday to grab an organism to determine the proteomics, the genomics, et cetera, and to have another host and to have a probability that these two organisms, if they interact, the outcome may be predictable, will be a symbiosis, will be virulence, et cetera. My next to last slide is I will tell you what I'm very proud about my lab. My lab took the mathematics and began timing my visits to the lab last summer. So in an entire laboratory effort, they analyzed when Arturo walked into the lab, they timed it. And then they measured the interval to the next time that I came in. And they were able to show for ordinals, there is a chaos. In other words, my visits to lab show chaotic behavior. I don't know where the chaos is coming from, but clearly there is a chaos generator someplace in my life that translates as a readout on my visits to the laboratory. So I promise to go back to the questions I posed to you. So why are we here? Why are we sitting here and not some sort of reptilian, or why are not a bunch of crocodiles attending a genomics meeting or something like that? The KT event, and there was fungal selection. Something had to happen that we didn't have a second reptilian age. The reptiles are very fit. They should have re-inherited the Earth, but something kept them down. And this group of organisms that was going nowhere, all of a sudden managed to become the dominant animals in the world. Why are we so hot? To keep the fungi away. Most of you eat all the time. Go to the gym and see how hard it is to burn calories. All that food is going to keep your pumps to maintaining a temperature way, way above the ambient temperature. And that is creating a thermal restrictive zone that allows you not to fear the fungal world. Why do you eat so much? To maintain temperature. And why are mammals the dominant animals? Fungal selection kept down the reptiles. There was no second age of the reptiles. So I want to acknowledge my lab, and I want to acknowledge the unpublished data that I presented to you. Quigley Dragotakis is a graduate student. Sarah Fu is a postdoc. They've been working on the KOC system. Nicole Broderick at UConn. It's been done Drosophila. Aviv Bergman, Albert Einstein has done the mathematics and has been the brains between all of this. Thank you very much. Yeah. And I finish on time.
With a lot of time for questions, if there are any.
Yeah.
With the temperature hypothesis,
Wait for the mic.
Wait for the mic.
thank you. With the effectively temperature exclusion hypothesis,
Right.
would you expect to see already much greater adaptation of fungal to higher temperatures in the tropics and equatorial regions, and therefore more pathogenicity?
That is correct, and that is the case. Most fungal diseases, most pathogenic fungi today, the species are found near the equator. Presumably because it's hotter and many of them have adapted and they have all the other pieces.
And you mentioned bats, when they hibernate, become susceptible. Are there other hibernating mammals like bears that
You know, I've been asked that question, but imagine trying to get the answer to that question. Who's going to go into a bear den when some of that bear is hibernating
an awesome field study.
to find if they got a fungal disease or to deal with the carcass? The answer is no. But there is another experiment, and that is that not all mammals are advanced mammals. So, for example, the monotremes, which are early mammals, they have a much lower temperature. They are quite susceptible to fungal diseases. So I think frogs have great BNT cells, and you have great BNT cells, but they don't have the thermal dimension.
Cool.
There's one in the back.
There's one in the back.
Oh, it's on. Okay. How do you suggest we develop antifungal therapies given that the genomes of fungi are so similar to our own?
So in infectious diseases, one of the great problems is that we have very few antifungal agents. And the reason for that is because at the biochemistry level, the animalia and fungi are very, very similar. In fact, but there are differences. They have cell walls, they use ergosterol, and they have differences in lysine metabolism. So there are sufficient differences that at least we have 3 major classes of antifungal agents. But in compared to the number of targets in the bacterial or parasite world, we are quite limited. Thank you. I think somebody there. Well, thank you very much. Have a good trip, and I hope that I gave you something to think about when you eat next time. Bye-bye.
Okay. Boy, we've done it. We're through. What a great week. Respect the fungi. I guess that's something else. Okay, I've got a few people to thank. Just want to wrap up here. Obviously, thanks to all of our speakers and poster presenters all week. It's just been amazing. Just amazing science. And just terrific presentations. I'd like to thank all the session chairs for keeping everything on time and steering us through a pretty extensive program this week. I'd like to thank the mic runners and all the other people that have been supporting the symposium and the main session. And thank all of the JGI admin staff for really putting on a terrific meeting this week. We're already going to start to plan next year's meeting next week, okay? We would love to hear all of your thoughts about what you'd like to see in the program, how you'd like to see the meeting. We have a survey that is live, I think, or about to go live on the app for the meeting. We're also going to send the survey out by email because we know not everybody downloaded the app. So we're really interested in your feedback. You can also shoot me an email. You can find out how to get a hold of me on the JGI page. I'm really interested to hear all your ideas. Just a reminder, our CSP call is now open and will be open for another 6 days. Letters of intent are due next Thursday, so please get those in. We'd love to have a great portfolio of projects with you all in the next year. And then next year's meeting, we are going to move to Oakland. It's great. We actually got the picture up there. And the dates, we really would love to see you all there. We promise it's going to be another exciting program. We can guarantee that. It'll be our 15th meeting. So thank you all for coming. Travel safely home. Have a great weekend. And yeah, that's it. Thanks, everybody.